teremos a participação, a apresentação será feita por Kaspar. Ele é bioestatístico da Roche e antes de ingressar na empresa, ele recebeu treinamento e trabalhou como estatístico nas universidades de Berna, Stanford e Zurich. Ele trabalha com pesquisa metodológica e dá consultoria estatística a equipes de projetos. Treinamento de bioestatística para estatísticos e não estatísticos, internos e externos. Ele interage com parceiros externos também da indústria, agências reguladoras e comunidades acadêmicas em vários grupos de trabalho e colaboração. Foi cofundador e colidera o Grupo Europeu de Interesse Especial em Estimates in Oncology, patrocinado pelo PSI, Statistician in Pharmaceutical Industry, e FSPI, European Federation of Statisticians in the Pharmaceutical Industry, que também tem o status de grupo de trabalho científico da ASA, American Statistical Association. Os interesses de pesquisa de Kaspar são métodos para otimizar projetos de estudo, análise avançada de sobrevivência, probabilidade de sucesso, estimativas e inferência casual, estimativas dos efeitos do tratamento em subgrupos e estatísticas não paramétricas gerais. Kaspar gentilmente concordou em fornecer seus contatos para qualquer necessidade após o evento, com acesso direto a contato a ele. Então, nós forneceremos para vocês. A recomendação hoje que temos, queremos testar uma nova forma na sessão de perguntas e respostas, então teremos 3 sessões de acordo com a agenda. Uma logo após o término do item 2, tá? De aproximadamente 7 minutos, depois uma segunda após o item 3 e uma terceira sessão de perguntas e respostas ao final da apresentação. O chat estará aberto durante toda a apresentação. Pedimos que, preferencialmente, vocês nos enviem as perguntas através do perguntas e respostas. Ele está ali, onde tem, logo ao lado do chat, tem os 3 pontinhos. Se vocês clicarem, vocês encontram perguntas e respostas ali. Tá? Antes de começarmos também, gostaríamos de solicitar a autorização de vocês para gravar essa sessão de hoje. Se alguém tiver alguma coisa que discorde, por favor, nos avise. E essa apresentação depois será disponibilizada no site da Interfarma para todos vocês. Então, eu agradeço desde já, que tenhamos um ótimo evento, que seja muito proveitoso a todos. Eu vou passar, então, a palavra para o Kaspar. Kaspar, thank you so much. We have just made the introductions. You feel free to start anytime. Thank you.
Thank you, Luciana, for the introduction. And I've been introduced in quite a few languages, even Chinese, but never in Portuguese before. So that was a first for me today. I would like to thank Interfarma and everybody who was involved to make this possible, to be able to talk about implications of the ICH-E9 addendum and how we develop, run, and analyze clinical trials. And one of the key messages of the ICH-E9 addendum is about collaboration. It's about collaboration between functions within pharma, statisticians, clinicians, regulatory colleagues, but also across companies in developing new methods, new guidelines, collaborating with all the stakeholders, because the estimand addendum tries to structure the dialogue with all the stakeholders involved: regulators, payers, patients, physicians, everybody involved in getting drugs to patients. So I'm very happy about this collaborative effort here today. Should you have any questions at any time, I invite you to just type them in the chat. I just need to bring up the chat again. So, okay. I'm just trying. Please type questions in the chat anytime. You hopefully should see that. So please type questions in the chat anytime. And then as I go, I try to address them, or we can address them during the breaks that Luciana has announced that we will have after sections. You can also just unmute yourself and ask a question. Okay. So I always try to make these kind of events interactive. And if we don't get to the end of the slide deck at the end, we maybe skip a few slides, that's okay. I think interaction is very, very important. So please ask your questions, bring up your case studies, contradict what I say. I think that makes for the most interesting discussions. Okay, so I would like to acknowledge a colleague at Roche from whom I borrowed the Yosemite example that we will discuss later today. And I have been involved in these estimate discussions for many, many years now. I have done a lot of research. I have implemented it within our company. I have worked across industry and with many regulatory agencies on making this concrete. And I would like to acknowledge many of these discussions. So this is not, a lot of what you hear today has evolved out of all these discussions. Here's a little agenda. So we will start off with a discussion of a few endpoints, mainly targeting oncology trials, to just set the scene a little bit. And then we will start to embed endpoints into the estimate framework, because endpoints is just one of 5 attributes in the estimate framework. And we will make that concrete using 2 examples, one from diabetic macular edema and one discussing new anti-lymphoma therapy, NALT, in lymphoma. And I think the addendum was met with quite some amount of skepticism initially. But a lot of that skepticism actually went away when many people saw how the addendum allowed us to very quickly and very efficiently assess the impact of COVID-19 on our clinical trials. Because I think without the addendum, that would have been much more difficult. But having that kind of structured way of thinking about things, we were able to quickly assess what is the impact of COVID-19 on many of our trials and then take respective measures. So I would like to discuss that a little bit as well. I will spend 1 or 2 slides discussing the scope. So to which kind of trials, to which phases should we apply the estimate addendum, or is it mainly intended to be applied? And then hopefully we get time to discuss the implications for clinicians and safety scientists. And this holds true for industry and regulators at the same time. I think the implications are very comparable. And then I wrap up with some overall impact and then a few concluding thoughts. So let's start with endpoints. And I will introduce and very briefly discuss traditional time to event endpoints that we use and what are some of the pros and cons with some of those. When we refer to time to event endpoints, typically we mean that we have a time interval that we measure between 2 clearly defined clinical events. For example, from randomization to death. Very simple events, you can measure the time in between and use that as a measure of clinical benefit. Why do we call time to event endpoints out here? Because, and this is not something I will discuss at length today, but imagine you have a trial with 500 people or 500 patients, and you randomize all of them, and you're interested in overall survival, so the time until death. Now, if you would wait, and if you would have the capacity to wait until all the patients have died, you would have 500 measurements, okay, of time to death, of overall survival. And you could use the standard statistical methods that we're used to. You could compute the mean or the median. You could use a box plot to display that data. You could compare 2 groups using some mean comparison. So very standard, very simple methods to analyze that data. The problem or the challenge or the difference with time to event endpoints is that you actually don't want to wait until all patients have died, because that could take forever. That could take a very long time. You might actually have a drug that cures patients. So what do you do then? You can actually lose patients. I think we're used to that. Patient at every time, at any time point in the trial, has the right to just say, I moved away to another country, I don't want to be part of the trial anymore. But as a matter of fact, when the patient leaves the trial, the patient's still alive. So what would you do then? This is what we call censoring. So censoring happens for these kind of endpoints. And that makes them special and does not just generate usual continuous data where we always have a number for every patient. Here we have a number, but we also have a status, still alive or dead. And that requires different statistical or specialized statistical methods, which we call survival analysis. Okay. As it is in oncology, many of the key endpoints are of this type 2 event type. So they all require this survival analysis techniques. And you might have come across this. So whenever you see Kaplan-Meier estimates, Cox regression, hazard ratios, these are tools that we use to analyze data that is censored. Okay, time to event endpoints. As we will see, overall survival might take a long, long time to be analyzable, even if you don't wait until everybody has died. Even if you say, I need 250 events to have 80% power, you still need to wait until you have 250 events out of 500 patients. So that might take a very long time depending on the disease indication. So there is an attempt and the desire to develop endpoints that allow an earlier decision whether the new treatment works better than the standard of care or the existing one. That's what we call surrogate endpoints. In oncology, think of response proportion. So you assess whether the patient responded to the initial treatment after 3 months or 6 months, and then you take that as an indication of treatment benefit. Pathological complete response in breast cancer is another surrogate endpoint, or minimal residual disease, MRD, in lymphoma, where you don't find any cancer anymore, and then you say, okay, or you cannot detect any cancer anymore. Then you say the patient is an MRD responder, and that might be associated with prolonged survival, but that needs to be shown. That's not a given. Okay, although biologically it might make sense, you still need to show that also statistically. So let's start with, in some sense, the easiest in terms of definition of these time to event endpoints, which is overall survival. That's typically randomization to death of any cause. We don't differentiate. If you're in a cancer trial, you die in a car accident, you are an event in that trial. Okay? You are assigned as an event in the arm you were randomized to. And it's clear, if we prolong survival, I think this is one of the key goals we have with treating cancer patients. So you want to improve how the patient feels, functions, or survives. And here it's about survival. A big advantage of overall survival is that it is objective, and you can determine the day on which the patient has died. So the investigator cannot influence the assessment of the endpoint. That even potentially allows not to blind the trial, potentially. OS also has some disadvantages. As I mentioned, typically we have a very broad definition of an event for overall survival. So it also includes deaths that are unrelated to cancer. And just because we don't want to enter that discussion, is it now related to cancer, yes or no? That you just avoid that discussion if you say all deaths are deaths, that's it. A difficulty is if you, for example, approve a drug on a benefit on progression-free survival, then the patients in the control arm might be tempted to switch to the newly approved treatment. This is something we call crossover. But if you then measure overall survival, you are not anymore measuring the effect of the initially randomized treatment A versus B, but you're actually measuring the effect of a treatment strategy. I start with the control therapy. When I progress, I get the new treatment. And that's a different comparison. And that's actually something that is to the core of the estimand addendum, because there one of the attributes is to define the treatment strategy. And I think we have not been careful enough in the past doing that, and now we are learning to do that better. It is very difficult, even with wonder drugs, to prolong overall survival. So you have typically not that large of effects, which means you need a large number of events to show small effects. If you have a small effect, you need more data to show a smaller effect. So that can make trials very long. You need a large number of events. And on top of that, in many indications today, we have large medians. So it takes long to observe. Good for the patients. It takes long to observe enough deaths in a statistical sense to be able to prove an effect. So that often makes it very challenging to plan trials with overall survival, to the extent that they become infeasible. And that explains the desire to have surrogate endpoints. So what are alternatives? Progression-free survival is one. And here I have some general remarks. I will not go into these. This is something you could potentially use in advanced disease. And then you also count progressions as an event. So on top, if you have the 500 patient trial, on top of those who die, also those who have a progression are counted an event for your time to event endpoint. Okay? And that means in the same amount of time, you will have more events. You will observe more events because many patients will have a progression and maybe die just a few years later. For overall survival, you would have to wait until they die. And now you also say progression means the initially assigned treatment has failed. And I have observed my endpoint. There is, I think, debate, and it's indication specific. Is PFS a surrogate for overall survival? Or is PFS, the absence of disease progression or death, a value in itself? Is it a value in itself for the patient to be disease-free? I think this, as I said, it depends on the indication. It depends on who you ask. I think there are differing opinions about the actual value of PFS. One advantage as opposed to overall survival is what we've discussed. Typically, the patients will remain on their initially assigned treatment until their first progression. So you don't have this challenge of patients crossing over after they progress. That's PFS. And then we have many other endpoints, and some of them are used in trials with a regulatory intent. Others are more used in maybe an academic environment. So time to progression, I think that's rarely used in pivotal clinical trials for drug approval. Time to treatment failure equally. Event-free survival, maybe a little bit more often. If you have a highly toxic therapy, it's not just about prolonging the time to progression or death, because if the patients cannot tolerate that highly toxic therapy, nothing is gained. So that means that you would also count discontinuation of treatment or when patients start a new treatment as an event, because it's basically a failure of the initially assigned therapy. Okay. So you see here the discussion about which endpoint is meaningful depends very much on the clinical context and what you want to show. It's not like you just pick one, the one you like, or there is a recipe which one to pick. It needs a case-by-case discussion. Part of that discussion has been enshrined in guidelines, and then you try to follow the guidelines or try to make sure you are in line with guidelines. So just to mention that. One aspect, and I see the question in the chat. I will come back to that question, very interesting question. One aspect that we need to consider that sets overall survival apart from virtually all other endpoints is death is very objective. But once we start, even with PFS, progression is something the doctor decides or makes a decision about. Of course, with using images and trying to objectivize with kind of RECIST guidelines or whatever as much as possible, but it's still a subjective decision. And for EFS, it's even more subjective because it's also the doctor who decides on together with the patient about discontinuation of treatment, starting a new treatment, etc. And that typically necessitates blinding so that neither the doctor nor the patient know which treatment they're getting, so that their decision to stop the treatment, to discontinue, or the decision to decide on PD is not impacted by the knowledge of which treatment they're getting. It often also requires, especially for PFS, an independent response review. Even more so if you cannot blind. Sometimes you cannot blind because the treatment administration, for example, is different. One treatment is like once weekly and the other is just every 3 weeks. Then it's clear you cannot blind. But then typically what is required is an independent response review. So you would collect all the images in your trial, send them to an independent response review facility without telling them which treatment the patient is getting, and they would just look at the pictures and redo the whole response assessments. This is actually a huge chunk in many of our clinical trials. Imagine I was involved in a trial that had in lymphoma that had 1,200 patients, and every patient, as per protocol, had 12 response assessments. Now, the clinicians among us, I think they know how many images you actually generate with such a trial. And then you have to organize that those being sent to an independent review facility. And it's not us who has these images. They're scattered around 200 sites globally. So imagine the logistics behind that. And often that is a major chunk of the cost of a clinical trial. So asking for an independent response review is quickly done, but has huge implications for those who run the trial, who sponsor the trial. And then there is disease-free survival. That's in indications where you get the patient to a disease-free status, like with surgery or adjuvant treatment. And then you measure the time until the disease comes back or the patient dies. That's what we typically call disease-free survival. If you're interested to learn more about all these endpoints, there is an FDA guidance that has all the details, the pros and cons, when to use which, and what it would be acceptable, what you need to consider when you use a certain endpoint. And now to this question, how to manage crossover in a trial designed to prove overall survival. So this is to the core of the estimand addendum, because if it depends what your question is. Is your question to show progression-free survival? Then OS is kind of secondary importance. Okay? And then you're less concerned about that. Because if you accept that you file on PFS, then you know the crossover most likely will happen. Patients will be unblinded. Sometimes patients will also take treatment outside of the protocol because you have reached your primary endpoint. If you have as your primary goal that you want to show an OS effect, then you might want to specify in the protocol what are the treatment strategies that we compare. Is it, say, the existing drug is A and the new drug is B? Is it just I randomize A versus B, I define the treatment strategy, the treatment schedule, and patients have to remain on that strategy until they die? Then you compare A versus B for overall survival. That is one way of comparison. If B is better than A, I think that's the cleanest way of showing that. But you can also argue, I mean, this might not be ethical, might be difficult. I just randomize A versus B, and then I follow the patients, and when they progress, those in the arm A, they're allowed to switch to B, because once you have shown that B is better on PFS than A, it might be unethical to not give A patients the option to switch. But then you're not comparing A versus B anymore. You are comparing A up to progression, and then maybe if the patient wants, gets B versus B throughout. It's a different comparison, which might be okay. I think we just need clarity on what we compare. So that's one way to answer that question. The other answer is that there are statistical methods. And again, we are to the core of the estimate discussion, and maybe we can come back to that when we discuss estimates. Because you can argue progression is an intercurrent event, and then I need to say, what am I interested in? Am I interested in the situation that I just described, comparing A plus B after progression versus B? Then I compare treatment policies. Okay, I would apply a treatment policy approach to the intercurrent event of progression. Potentially, I could ask myself, I'm actually interested in the effect of A versus B in a world where progressions in arm A would not happen, where crossover after progressions in arm A would not happen. So I'm more interested in kind of the biological clean effect of A versus B. That's a valid question you can ask. And there are statistical methods you can correct for the fact that some patients cross over to the new treatment after progression. You basically replace them with patients that didn't cross over. But you need to make assumptions about what this crossover mechanism actually is so that you can model that crossover mechanism. You also need to collect a sufficient amount of data at the time of progression to be able to model that switch. But you can then answer that question in a hypothetical world where no crossover would happen. What would have been my effect of initially randomized A versus B? But you need to say what you're interested in upfront so that a trial can be planned accordingly. Okay. And the EU has, the EMA has a question and answer document precisely discussing that question, where they say, if you want to kind of use statistical modeling to, in some sense, remove the crossover effect, then here is a list of what you should think about. And that was a result of this whole estimate discussion, that we now have kind of at least in theory, a path how we could account for crossover also in regulatory interactions. Because in the past, that was some kind of a no-go. You would just measure overall survival irrespective of what happened to the patient. Although we have cases where 80% of control arm patients crossed over to the new treatment. So if you then just measure OS and you don't see a difference, I think, or not a big enough difference, I think that's not so much of a surprise. I just shamelessly plug here, the Oncology Estimate Working Group has written a paper on crossover. So if you're interested to learn more about this, I invite you to go to our webpage, oncoestimate.org, and there we have many presentations discussing precisely that question and what you can learn from one way or the other. Or you reach out to me if you want to know more. So I pause here. That was, I think, oh no, not yet. So I think we still continue to give an intro into estimates, but maybe there are more questions in the chat, which I invite Luciana or someone else to translate so that I can maybe provide some feedback.
I cannot see any questions so far. It's just instructions here in Portuguese, Kaspar, for the Q&A.
Oh, okay. So I send you the question.
Yeah, maybe. Okay. Maybe a question was just sent to you and we cannot see.
You see them now?
Let's see.
I think one question is about whether I'm able to share the slides, and if for me that's not a problem. If you want to share the slides to those who attend today, that's perfectly okay. So that I think was one question. but I could just see PDF. So I think that's what I guessed
Yeah,
that
this
from.
is, you have translated perfectly well. This is what is being asked. It's going to be shared with the audience.
Okay. Any other questions? You can also just unmute yourself and ask questions. Okay, so maybe we continue for a few more minutes and then we have a formal break and then we can open the floor for discussions. So we have an FDA guidance. We also have an EMA anti-cancer guidance. We have all these perfect definitions. It seems this is all clear cut. It's easy. Okay, that's what we thought for many decades. Until suddenly and slowly, people started to think about things and consider and start to ask, do we really know what we're doing with all these endpoints? Here are a few counterexamples. Disease-free survival in breast cancer. This is a paper, it's about 15 years old already, where a group sat together and then they extracted the information from 5 major clinical trials that had been run in this indication. Because this is a very important endpoint in breast cancer. And typically in adjuvant breast cancer, this is the primary endpoint you choose. And very often, we all know how this works. You have a trial, and then you have a new trial, and maybe another trial, and then you compare across trials. You find differences, and then we discuss at length what made the difference. Maybe it's just the endpoint definition. So what this group did, they took these 5 trials, all huge, large trials, collaborative group trials or industry trials. And then they sat down and precisely looked into how was the endpoint actually computed. And here you see the trials. Each row is a trial, and each column is a clinical event. Okay. So if you have a local or regional recurrence, of course, for DFS, you would think that's an event. And that was actually the case in all of the trials. If you have a distant metastasis, that's also recurrence of the disease, of course. So it's an event for all the trials. If you die from any cause, you see 2 trials, they didn't count that as an event. It's quite interesting. You would think DFS is, you would consider death an event because even maybe you haven't measured the recurrence or you haven't diagnosed the recurrence, but if a patient dies with a history of cancer, I think it makes sense to count that an event, but only 3 trials actually did. Here's another, I leave that to the clinicians, what exactly that is, but another clinical event that all trials counted an event. But then you see here, second primary invasive cancer, non-breast. Here they disagreed again. And then here you have further clinical events that none of these trials treated the same way. So if you see differences between results in these trials, maybe they share the control arm, but the survival functions, the Kaplan-Meier estimates for the control arm are quite different between the trials. And then we start to discuss at length what might be the cause, is a different population, whatever, I don't know. You see, the devil can be pretty much in the details, namely how the endpoint was computed. And none of these 5 trials have precisely the same definition of endpoint, which is quite worrisome. So this, I think, so this is how I kind of 15 years ago started to think about, are we kind of stringent enough? Are we kind of precise enough in what we do? And I think the estimand addendum was then ultimately a culmination of all these thinking of are we kind of precise enough? Are we doing things right? So that should be a kind of a warning example that they all said, if you look at these papers, all published in the best journals, they all say DFS is our primary endpoint. But DFS is not the same as DFS potentially. Similar for PFS. This was the trial I was mentioning. I was the trial statistician for 5 years of the gallium trial that led to the approval of Gazyva in follicular lymphoma. Okay. And here you read the endpoint definition. The primary endpoint was progression-free survival as determined on the basis of the assessment of tumors at an independent review facility. And then PFS was defined as time from randomization to first evidence of progression or death. Okay. Pretty simple. That's what we discussed, progression or death. But what happens? So that's the ideal case. You randomize, you start the treatment, and then you wait until the patient either progresses or dies, and that's your event. And those who didn't die and didn't progress, you would censor at the last response assessment, because the last non-progressive response assessment is the last time you know the patient didn't progress or didn't die. Okay, so that's the definition of censoring. The last time you knew the patient didn't experience the event yet. But this trial had 1,200 patients randomized. And before unblinding, the clinicians suddenly started to discuss, well, and you don't need to be unblinded. You can just 1,200 patients and you can look which patients actually received new anti-lymphoma therapy before progression. Now, say 10% of patients, you do that blinded, 10% of patients have received such new anti-lymphoma therapy. If this is all in the control arm and they get a highly effective therapy, that puts at risk your ability to show an effect in that trial, of course. If the patients need that therapy, that's perfectly okay. That's not something we should try to organize away with a very restrictive protocol. That's not the point. The point is that we should upfront say what we do with these patients that get new anti-lymphoma therapy before they progress. Okay. We need to have that discussion upfront. About 2 approaches come to mind. Either you can say, I count this as an event because it is indicative of a treatment failure, but then your endpoint is actually not PFS, but EFS, event-free survival. The other approach is you say, I compare the initially randomized A versus B, Gazyva versus rituximab, and if in one arm, patients get a lot of these new anti-lymphoma therapies, it's just indicative of the failure of the treatment, and then we will not show a PFS effect. But that's exactly what's happening, because if they all need this new anti-lymphoma treatment, it's this initially assigned treatment is no good. That's another strategy. So either you count it an event, or you just ignore it and say, it belongs to my treatment strategy. Okay. And these are 2 of 5 proposals in the ICH-E9 addendum, how to handle such an intercurrent event. We will discuss what exactly that is later. Either you make it a composite, you make it a part of the endpoint, or you just say it's part of my treatment strategy. Either way has arguments in favor or against. And it's not the point whether one is better or than the other. The point is you need to discuss this up front. Okay? You need to be clear, what do we do with these kind of patients? That's one of the key implications of the ICH-E9 event. And of course, I think we all agree the interpretation of a PFS difference between Gazyva and rituximab, this interpretation is different depending on whether part of the patients received new anti-lymphoma treatment before progression, yes or no. And the question what to do with these patients is not one way is better or the other. It's just what objective do you have? And we will discuss what objective actually corresponds to the 2 strategies that I was just outlining. So upfront discussion is important between all involved stakeholders. This is not something the statistician can figure out. This is something the statistician can account for and describe and be precise about. but only in an interaction with the clinician. And just to illustrate that the same word might mean very different things, we also have drug development programs in hemophilia. And for a long time, the standard endpoint in hemophilia was annualized bleeding rate. So basically, you just counted how often these patients had a bleed event, and if they had, like, I don't know, 10 in half a year, you would say the annualized bleeding rate is 20, because then if they had been followed up for a year, they would have had 20 on average, just extrapolating. And you would call that an annualized bleeding rate. And you see here a couple of papers again, so it's a very similar exercise compared to what I showed for DFS. And the primary endpoint was called ABR in all of these trials. Same name. But you see here, none of these trials use the same statistical method. And that makes kind of comparability very, very difficult. It's even the question, are you measuring the same thing? Are you comparing it in the same way? Just to illustrate again that just by using these broad labels, PFS, EFS, ABR, it's just not precise enough. You can still understand something very different under that. And this is all not new. There is a paper in 1977 by Pito and colleagues where they give recommendations how to define and report time to event endpoints, but that was grossly ignored. Altman, one of the godfathers of clinical statistics or medical statistics, picked that up in 1995, finds frequent failure to specify whether non-cancer death treated as events or censored. Reiterate the need for clear definition. That was 95. 2008, half of reviewed articles in major clinical journals failed to provide clear endpoint definition. So we're not learning, it seems. 70% reported insufficient information on survival analysis. 5 years later, most of these time to event endpoints lack standardized definition. And depending on the definition, I mean, if you go back here, I think it's easy to construct examples where one definition would give you a statistically significant trial and another definition would give you a non-significant trial. That's not what you want. And that is kind of illustrated in this publication here. So you see here, quite a famous trial in colon cancer. Depending on the definition you apply, you get a significant or a non-significant trial. So there's something to be done about this. And there are efforts in the direction of unifying these endpoints. DateCan is one initiative that brings key opinion leaders together, and they sit together for a week, and then they discuss what is this endpoint, and they generate very specific guidelines. Which clinical event has to be counted an event for that endpoint, or ignored, or censored, or whatever. So this has been recognized. But the estimand addendum is another step in just trying to be more precise what we are doing and make things more comparable and make more clear. And I just want to iterate, this is not a challenge specific to oncology. I think it applies to all endpoint types and all indications. Even for response, a seemingly simple endpoint, binary, you find different definitions. And it's just not helping. I think we need to make sure things are more aligned. Okay. Again, I just stop here for any questions you might have.
Any questions from the audience? You can include in the question and answers or in the chat. I don't see any new questions here, Kaspar.
Okay, so then we move on to section 2, and then after that we will have a 7-minute break.
Correct.
Okay, so what's the estimand addendum about then? ICH, International Council for Harmonization, has issued E9 in 1998, which is quite an achievement. E9 kind of bundles key statistical considerations, how to run clinical trials. And for more than 20 years, it was found to be useful and sufficient for what we do. But a couple of years back, and what I just described in terms of endpoints was just one aspect. There were other aspects where we just kind of, we focused too much on analysis methods. For example, when you think of missing data, we discussed a lot about which method should we use to account for missing data, MMRM or something else. And we somehow lost track of what are these methods actually estimating? What are the underlying assumptions? We were focused a lot on the analytical side and less so on the conceptual side and thinking about what is the objective we want to achieve with a given trial? And then the objective defines the analytical method. Very often it was actually the other way around. So the trial objectives, it was found, were not sufficiently precisely described. And very often we just did some favorite analytical method, and that method implied a trial objective or a question you answered. But not everybody was clear about that question we answered with a given analytical method. And if they had known, they would actually have not agreed to the question we answered. So there was some kind of disconnect between the analytical methods and the trial objectives and how we quantify treatment effects. And it was felt that we need to fix that. We need to be more specific and more precise in description of treatment effects and the analytical methods we use to estimate them. And the estimand addendum introduces 5 attributes that we need to describe. In the past, I think we focused a lot on endpoints. And even there, we somehow failed in some sense, when you remember all these discrepancies and heterogeneity that I illustrated. The addendum adds further aspects that need to be specified when we describe a trial or a treatment effect. And you see here, so you want alignment between the trial objective and the objective we have typically in many of our trials is we want to show that drug X is better than drug Y in efficacy or safety in indication Z. I mean, it's no surprise that that leaves a lot open. It's very imprecise. It's very high level. And I think it makes sense to be more precise there because different stakeholders may understand different things about such a high level objective. And there are many examples where that different understanding for different stakeholders only surfaced when you had already collected the data and went for a filing. And then at the filing time, the regulator said, we thought you do A. And the company said, well, we thought we do B, but A is not the same as B. So the estimand addendum has the ambition to generate alignment between objectives, the trial design, and the data collection, because depending on how you describe all the attributes, you need to collect different kinds of data. For example, if you want to account for treatment switching, you need to collect data at the switch to model the switch. If you say, I don't care whether patients switch, I just measure OS, then you don't need to collect that data at switch. And I think it's very helpful to be clear upfront about that feature. Okay. The addendum also introduces a clear definition of a treatment effect. It clarifies what is a sensitivity analysis and what is a supplementary analysis. So sensitivity analysis is targeting the same estimand, just varies the statistical assumptions. And the supplementary analysis is targeting a different estimand. So I think later we will see the difference. It's also a promise to, very often we have kind of this broad range of sensitivity and supplementary analysis. We just vary all kinds of stuff in our analytical approach without actually being very clear. Why are we varying all these things? And then we have maybe 15 sensitivity analysis. If they all go in the same direction, we're all happy, pat on our shoulders and say, okay, that's fine. But if 4 out of 15 go in a different direction, what is then? What does that mean? I think we were not clear in the past about what to do then. And then we just try to discuss these 4 away and say, oh, we shouldn't have looked at this anyway. But that doesn't seem to make a lot of sense, does it? And the addendum tries to fix that. Maybe you just have 3 sensitivity analyses, but it's very clear what their purpose is and how to interpret them up front. A very important aspect of the addendum are intercurrent events. And what is an intercurrent event? That is a clinical event that happens between randomization and your endpoint that may alter the interpretation of your endpoint. So if you think of MALT for PFS, I think if a patient gets new anti-lymphoma therapy before progression, that alters our interpretation what PFS means for that patient. And it's not about a good or a bad way to handle that. I think that the point is we should think upfront about what do we do with patients who have such an intercurrent event of MALT. And you may have a long list of intercurrent events, and for each and every one, you should say what to do with that intercurrent event for that patient. Okay. So you see here, one important aspect of the S-demand addendum is a clear definition of a treatment effect. And maybe I could pause here and have you think, what do you think is a treatment effect? It's actually a very difficult concept. Are you, in your head, are you clear what is a treatment effect? It's not obvious. And interestingly, while E9 is the efficacy guideline, in its initial version, it doesn't define what a treatment effect is, which is, in hindsight, is quite surprising. You would think, I mean, efficacy is about treatment effects. So what would be the first thing you would expect in a guideline about efficacy? Is a definition of a treatment effect, but it's not there, okay? So what is a treatment effect? Well, a treatment effect asks for some mental gymnastics. Because how we define a treatment effect is using what we call in statistics potential outcomes. So you want to compare drug red against drug yellow. A treatment effect is defined as assuming that you have a patient and the patient would get drug red and have some effect. Then you would revert back in time and take the same patient and give the same patient drug yellow. Then the patient would again have some value of the endpoint. This can be overall survival. Of course, you cannot do that for a patient. The patient can only just experience one drug, unless you have a chronic disease that reverts back if not treated, etc. I mean, that basically the situations where you can do crossover trials, that can happen, but typically that's not what we have. So you would, the thought experiment is the patient first gets drug red, has some endpoint value, and then gets drug yellow and has some other endpoint value. And the difference here is the treatment effect. So that's a metaphysical thought experiment because you cannot revert back in time. So how do we get to that treatment effect? Well, we can estimate it from a randomized trial. Because in a randomized trial, 500 patients, 250 get drug red and 250 get drug yellow. And at randomization, their distribution of potential outcome here under red and under yellow is the same. That's what you achieve with randomization. Their potential overall survival is the same at baseline because you pick them from the same, I mean, on average in the 250 and 250, okay? So if you find the difference between overall survival between this group and this group at the end of your trial, it must be the treatment. And with this randomized trial setup, you actually estimate under certain assumptions this average treatment effect that you define here using this metaphysical potential outcome experiment. And that illustrates why randomized trials are so important, because it's the simplest device to estimate such an average treatment effect. If you don't randomize, say you just pick 250 and 250 and the doctor decides who gets which treatment, then your distribution of the potential overall survival is actually not the same anymore in the two treatments. And you can mathematically correct for that using, for example, propensity scores, but then you need to put in assumptions. So either you have randomization with few assumptions, or you have non-randomized data plus assumptions, and then you can also estimate an average treatment effect. It's just a different way of estimating that average treatment effect. And that's now fixed in ICHE 9. ICHE 9 defines a treatment effect in this way. I think we skipped that. Well, okay, so the original E9 has this famous intention to treat principle. And what the intention to treat principle basically says, if you are randomized, you're just followed up to the endpoint, and irrespective of what happens in between, we just measure the endpoint, and that's it. Okay? So with this, you assess the effect of a treatment policy. So think of the treatment switching example. You assess the effect of a policy. You start with the control. If at progression, PFS has already been successful and you filed, you get the chance to switch to the new treatment, and then we measure overall survival. That's my treatment policy, and ITT measures the effect of that treatment policy. Okay, and that's also what the original ICHE 9 puts forward. But the problem is that's all simple. If you follow all patients up until they have overall survival until they die. But what do you do if a patient leaves the trial? If a patient, for example, discontinues treatment or has new anti-lymphoma treatment, ITT doesn't upfront tell you what to do necessarily. So how should we analyze the data from patients who did not complete the treatment as planned, and what effect are we then estimating? Because here you basically, this is the perfect world. If everybody is followed up, everybody completes treatment as in the protocol, then you can estimate that ITT, that treatment policy, treatment effect. But ICHE 9, the original guideline, is not very helpful when it comes to say, but what happens if some of the patients actually don't finish the treatment as pre-specified? Okay. That's just lacking there. And everybody was left alone, what to do then? And then I think that kind of introduced also this heterogeneity and this lack of upfront specification. And this might have very serious implications. So as one colleague from Roche put it, bad drugs might appear good and good drugs might appear bad, depending on what you do with these events that kind of prevent the clean observation of your endpoint here in this ideal world, which typically is never true. If you have 1,200 patients, it will not happen that 600 in arm A will finish their treatment as per protocol and be followed up until the endpoint, and 600 in the other arm will do the same. It will just not happen. So you need to say what to do. And that is one thing that addendum tries to fix. And it tries to fix that using this estimate definition, and I think we went through that already. It defines what treatment effect will be estimated, okay? And it does that by being very precise on 5 components of what a treatment effect is. And we will discuss these components, of course. Here are they. I think typically you start with the population. Be very precise what the population is. That's not very different from what we did before. The endpoint or variable, as the addendum calls it. Again, this is not so different as before. So you would say PFS or OS or EFS. One thing that is different is you are very specific about the treatment. And that might actually include background therapy, concomitant medications. What is my strategy? Is my strategy I get A, and then after progression, I'm free what I get? Or after progression, I need to get the treatment from the treatment arm. You just need to be very specific. What is your treatment strategy? And then I think one of the key novelties in the addendum are this definition of intercurrent events. So here you would say PFS, but then you would sit down, clinicians and everybody involved in the trial design, and start to think about what clinical events can happen between randomization and progression or death. And then you would start to discuss, when you design the trial, NOLT, for example, new anti-lymphoma therapy. Because obviously that alters the interpretation of your endpoint of PFS. And then you discuss what now to do with a patient who gets NOLT before progression. Are we interested in a world where NOLT would not be given? On certain circumstances that could be of interest, then you use certain methods. Do we say NOLT is indicative of treatment failure? Then you would just count it as part of your endpoint. Then you would change the endpoint to EFS. So you see, quite naturally, that sparks these discussions that are necessary. And we will discuss examples where this will become very clear, hopefully. And then ultimately, and I think that's also not so different from before, you specify a population level summary. For example, if you have PFS or OS, you would say we compare the groups via a hazard ratio. Or if you have non-proportional hazards, you use something else. And I think these are more statistical aspects that we are pretty versed in dealing with. So, and then all these aspects make your estimate and are precisely defining your trial objective. Okay, and you see this is very different from just saying we want to assess whether drug A versus drug B is improving efficacy and safety in indication X. Here you are much more precise. And then the hope is that brings a lot of the discussions we had at filing time to the design stage, and we can clarify that every stakeholder has the same understanding, and that once we agreed on an estimate, we can set up the data collection strategy such that we can answer the questions that need to be answered for that estimate. So that's a lot of stuff, I'm aware, and I'm more than happy to pause here, and I'm curious to see whether there are any questions or comments.
Perguntas da audiência, tivemos uma apresentação bastante densa, né? Acredito que tem alguma pergunta por aí. No questions, amazing presentation, congratulations. Kaspar, this is for you. Let's see if anybody else has a question. Okay. Questions coming. The first one is, how do you see the use of estimates to work with real-world data?
Very interesting. And so the estimate addendum has in mind clinical trials, but it says in the addendum, whenever you are interested in a treatment effect, the structure and the framework put forward in the addendum is useful. And I think that's true. So we also apply it to questions in real world data. Because also when you have real world data, you are interested in estimating a treatment. Often you also use real-world data to inform other aspects, for example, assumptions in a clinical trial, then you are not interested in the treatment effect. But if you are interested to use real-world data to estimate the treatment effect, for example, contextualize a small trial in a rare disease, you are interested in estimating a treatment effect. So the concept applies. And for those of you who are a little bit familiar with what's going on in the statistical world, the way I define the treatment effect here is kind of a causal treatment effect definition. And this is what we are after typically. We want to estimate the causal treatment effect. If I change from A to B, what will happen to the patient? And these are typically also the questions for real-world data. You have these causal questions. The fact, and then I think we need to discuss a little bit. There are various degrees of real-world data. But in general, the answer is, or my answer is, yes, the framework is equally well applicable to real-world data. And actually, in the oncology estimate working group, we have a sub-team that is looking to apply the estimate framework to questions for real-world data. Just to get more clarity about what we're doing, just to put transparency on the assumptions that we are making when we do some kind of analysis. Also internally, we have an initiative ongoing with Flatiron where we precisely use the estimate framework to better understand certain analysis that we do.
Okay, so Kaspar, we have another question. What is the best approach to address with the intercurrent events in COVID-19?
So I will discuss that when we have time. I think it depends that there is no uniform answer to that question, in my opinion. It depends on the indication, and it depends on the type of intercurrent event. And the Oncology Estimate Working Group has written a paper considering this for oncology. And we made one key distinction we made is when we said there are intercurrent events happening because of COVID-19 that are completely independent of the patient and the disease and the treatment. For example, if your local hospital is shut down, that's completely independent of your disease, of your status, of your treatment. There, you could argue that I remain interested in a treatment effect in a world where that hospital would not have shut down because of COVID-19. Because at some point the pandemic will be over. And I remain interested in the treatment effect in a world without the pandemic. Because the drug will potentially be used for decades to come when there is no pandemic. Okay, so everything that is induced through the pandemic, but which is completely unrelated to the treatment or to the patient, you can argue you use a strategy to address that intercurrent event of, for example, the patient discontinuing treatment. That would be the intercurrent event, discontinuation of treatment due to closure of the hospital. There you could apply a hypothetical strategy. I think that makes a lot of sense. You can argue it's different if the patient discontinues treatment due to a COVID-19 infection, for example, because there you can never exclude, although maybe everybody says it's because of the COVID-19 infection, but maybe the patient would also have discontinued without the COVID-19 infection because he was just not doing very well on the treatment because of the disease. So here you can actually not really make a distinction between, is it now COVID-19 or is it the disease or is it the treatment that makes the patient discontinue? So here you can argue maybe a treatment policy strategy makes more sense, so that you just kind of treat that patient as if he or she had discontinued due to any other reason. If you're interested to learn more, there are at least 2 papers that discuss this quite extensively how you can deal with certain intercurrent events due to COVID-19. But I think it's indication specific. Oncology is different than neuroscience, is different than think asthma. I mean, that's even more complicated because COVID-19 is also a pulmonary disease potentially, or a disease in kind of the same indication. So I think it needs a case-by-case assessment per indication, I would say. But yeah, very relevant question, and that kept us busy, and it still keeps us busy for more than a year now to assess the impact of the pandemic. Yeah.
Thank you, Kaspar. So I make one more question in the interest of time, okay, and we can go back later at the end. So why the forest plot evaluations are not mandatory to show the interaction tests among subgroups?
Can you repeat the question again?
Why the forest plot evaluations, okay, it's not mandatory, they need to show the interaction test among subgroups?
I don't know, mandatory. I think we, in our standard outputs, we even add the interaction test, I would think, or we provide a table with it. I think for that question, we need to think of what is the purpose of these forest plots that we show. And I think my sense is the purpose of these forest plots is to assess whether the treatment effect is approximately homogeneous over all subgroups. And for that, I consider kind of the broad picture. If you have overall a treatment effect, but then in every subgroup, it's all over the place, then that's kind of strange, okay? But if you have mostly homogeneous effects, and maybe there's one or the other subgroup where it's a little bit different, I wouldn't bother so much. So that's for me the purpose of a forest plot. For me, the purpose of a forest plot is actually not to find those subgroups where the treatment effect differs or is smaller. Because if you want to do that, you run in a lot of problems, multiplicity problems, pre-specification problems. If you just look at enough subgroups, you will find some that go in another direction. And while I think it's useful, the interaction test as maybe adding some explanatory tool, I'm not sure, or I think one would need to carefully think about the situation where the interaction test p-value would lead to a different decision than from what I see in the forest plot already anyway. But I mean, it's, yeah, I don't know whether there is a deeper reason that we don't add this by default, or some companies maybe don't add this by default. But I think there is a risk that we kind of overinterpret what we see, because even if you then have a p-value of an interaction test that is small, first of all, you would have seen that in the forest plot already anyway. And then you need to counterbalance that against how many subgroups did I actually look at, and not just focus on this very small p-value, the single one that you just have. So if you're interested to learn more, I mean, I have a whole course on subgroup analysis that I give internally, and I'm happy to share some more insights. There are quite a few publications who discuss this. We've even done research in this area, kind of adjusting effect estimates for the fact that you look into many subgroups. So if you're interested to understand more, then I'm more than happy to discuss. So just send me a message, and then I'm happy to share some material.
Okay, thank you, Kaspar. So Vichy, we can go back to the presentation, the examples now.
There's another question, but maybe I can answer that in the next break.
Yes,
that's okay. I keep that in the chat. Okay.
Okay, thank you.
Okay, so now we have seen this estimand addendum. So let's discuss a few examples, or two at least, or at least one, say. DME, diabetic macular edema. Okay. And just to say, or just to illustrate the ambiguity again, and I will not tell you much new stuff now, but I think it maybe is a different way of discussing the same aspect and just making it clear. The primary endpoint in the Yosemite trial, which is a trial to assess faricimab, before we implemented the estimand addendum, read approximately as follows. The difference between faricimab and aflibercept in change from baseline in best corrected visual acuity at 1 year, BCVA. But again, that leaves a lot of stuff open. And here is just a few things, and we discussed that so already. So some patients will actually adhere to the treatment perfectly and follow as according to protocol, but the green ones, they will not. Okay? They will not. They will just not adhere. Some patients will require additional medication. For example, here the orange ones. And the gray ones, they will not. Okay? Now, if you put that together, you can define different treatment effects. You can say, so here is the BCVA value at some time point, or the average at over 3 time points. You see it here. So those who don't adhere, they have a value of 60. And those who adhere, they have a value of 70. Now you can say one way of defining a treatment effect is just regardless of whether they adhered, yes or no. That's treatment policy. That's the same, or that's a comparable situation as of irregardless whether they switched treatment, yes or no. I just take what I have at the end. Okay. Then you would average these numbers here. You could also argue that I'm actually interested in the hypothetical condition that all subjects could adhere to the treatment. Then maybe those that couldn't adhere would have a value of 80. We don't know, but you can use statistical modeling to impute this value here. For those who didn't adhere, you just use some statistical model, and that gives you the hypothetical world if they had adhered, and then you would average these numbers. Okay. Or you can say, so that's then you would use a hypothetical estimand for the intercurrent event of adherence. Okay. Or you could argue, I'm actually interested in the strata of the population, and it's not a subgroup, it's a strata that can adhere to the treatment. So I just remove these patients, and that's actually not as per observed adherence here, so it's a little bit tricky, and that's the principal stratification strategy that is proposed in the addendum. Okay. So depending on what you're actually interested in here, you get different values. So here you ignore the fact that some don't adhere. Here you predict the outcome for those who cannot tolerate. And here you only use those who can tolerate. These are all perfectly fine scientific questions. That's not the point. The point is everybody should be on the same page which one question we are answering with this trial. Okay? And be clear up front. And you see here, you get different values because you're estimating a different thing. And what thing you're estimating is defined here. Okay? And the same is true for additional medication and any other intercurrent event that you can think of. For every of those intercurrent events, you need to choose a strategy. And there are actually 2 more in the addendum described, and maybe you can also come up with some more. It's not meant to be an exhaustive list. Okay? And this is this ambiguity. And it's not about saying there is this one way how you have to do things. That's not the point. The point is just be clear. Just be transparent about what you think you should do and what makes scientifically sense. Okay? So that's a clarification for intercurrent events. And now we can build the estimate for Yosemite. So the population are the subjects targeted by the scientific question. And you see here, adult patients with DME, blah, 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 blah. That's as defined. Typically, that's the population you define by your in- and exclusion criteria. Okay, so that's not something that's very different from what we've done before. This is the endpoint. Change in BCVA score from baseline, averaged over weeks, blah, blah, blah. Okay. Treatment. And here you are more specific than what we've used to in the past, what's been used to in the past. You specify, for example, the interval in which you're giving the treatment, etc. So this can look much larger or much longer for an extensive oncology treatment, for example. But that's the idea, that you're very precise. What is the treatment that I compare? And then the intercurrent events. So these are clinical events that occur after treatment initiation and either preclude the observation of the endpoint or variable. The addendum talks about variable, but it's the same as endpoint basically, or affect its interpretation. So treatment discontinuation, we have seen, use of systemic treatment that is not foreseen in the protocol. That always happens in clinical trials. There's no point in pretending it will not happen and not be clear about what should we do if it still happens. It will happen. In 1,000 patient trials, a lot of stuff happens. So we need to be clear up front what we do with that. Okay. The addendums gives you 5 strategies. We have seen quite a few already. Treatment policy, hypothetical, principal stratification, composite, and what else? There is one more. You just make it part of the treatment. So these are the, no, that's treatment policy. There's another one. So there are 5 strategies in the addendum. And the idea is that you align them with the clinical question of interest. And you can have, for one intercurrent event, you can have treatment policy. And for the other intercurrent event, you can have hypothetical. So it's not like you need to use the same strategy for every intercurrent event. So this is, you see, there is a lot more detail that I use to describe the treatment effect and the estimate that I'm interested in. Here we have 2, and the decision of the team was that you will use a treatment policy strategy. So you just ignore if patients discontinue the study treatment, you just measure BCVA at week 48, 52, 56 and average that irrespective of whether the patient discontinued the treatment, yes or no. Because you can argue if a patient discontinues treatment, it speaks against the treatment. It's not tolerable. So if then the score is bad at week 48, 52, 56, that's just a result of the treatment. And we should measure that, okay? And the same for if they need to use any prohibited systemic treatment therapy. Okay. Typically, treatment policy is the simplest strategy to apply because you just, for every patient, you just measure 48, 52, 56 and average, and that's it. But you need to be careful that you collect the data. So if a patient discontinues treatment, in the past, sometimes we just stopped collecting the data. But here you continue to collect the data because you will still use here what's happening at week 52. Okay, so even if the patient gets off treatment, you should make every effort to continue to collect the data. And then population level summary, that's kind of basically just the mean difference computed in some complicated way, accounting for covariance. So that would be the estimate for Yosemite. You can go through the same exercise for NOLT in lymphoma. I will not do that. So when you get the slides, you can have a look. Here, I relate that back to the objective of the trial. Sometimes a treatment policy strategy makes sense for NOLT, and sometimes a hypothetical strategy makes more sense for NOLT. But whether it makes sense depends on the trial objective that you try to achieve. Okay. You need to relate things back. What do I want? And that informs what do I do. And we should not start with, here is what I do, and then, oh, it implies this and this. It should be the other way around. First, I should be very clear, what do I want? And from that, I derive, what do I do? So why, how, and what? So that was a bit shorter, and maybe I can just start answering this question that's still in the chat. Regarding this.
Just a question to you. In the interest of time, what do you think? We have 30 minutes to the end. What would you prefer that we leave those? We have just a couple of questions because one I think it has been answered already. And do you want us to leave it to the end, these questions, or what do you think, considering what we are missing to the end?
I think we can answer a few questions now. It also for kind of
okay,
a little bit, not just me talking all the time. So maybe that's good. And I think it's we still have not so we are far beyond half of the material. So I think it's pretty okay.
Okay. So let's see. We have two questions regarding sensitivity. I'm not sure if they are related, but let me translate this one. So in regards to intercurrent events, making the sensitivity analysis would be enough to evaluate the treatment effect. Do you want to ask again?
Yeah, maybe I just missed the part. Can you repeat again? Or
so in regards to intercurrent events, okay, making the sensitivity analysis would be enough to evaluate the treatment effect?
So sensitivity analysis, so maybe I'm not precisely answering the question. I just try to give some context and maybe by chance answering the question eventually. Sensitivity analysis, the purpose is that we assess the impact of different statistical assumptions. Okay. So an easy example is for if you assess a mean difference. If you do a t-test, you make some assumptions. If you do some other test, you make different assumptions, but you're still targeting a mean difference. So your estimate is the same, but your statistical assumptions about how the data is distributed maybe is different. Okay, so that could be a sensitivity analysis. Intercurrent events, if you start to change the intercurrent events or the strategy with which you handle intercurrent events, you're actually changing the estimate. So if you start to treat intercurrent events differently, you're not targeting the same estimate anymore. So that would rather be a supplementary analysis. If you say, instead of using treatment policy for discontinuation of treatment, I use hypothetical. Then you change the estimate, and we would classify that as a supplementary analysis and not a sensitivity analysis anymore, because you change the thing you're estimating. You're not just changing the statistical assumption of your method. I'm not sure. I hope that kind of sheds a little bit of light on the question. Otherwise, just feel free to insist.
Okay, so Miriam, if your question was not answered, please send it again. And we have another one related to sensitive analysis. It's in the chat. I'm not sure if you want to read it.
So the fact to anticipate those parameters before the study start could not be precocious in some aspects, as we do need to review some of them depending on the study results. I'm not sure precocious. That's maybe lack of my English.
Maybe it's prevent.
Not
sure. Let's see. Yeah, it makes sense or no?
Maybe the question could be, what happens if we only find intercurrent events during the review or things that happened that we didn't pre-specify? I answer that question and then let me know if that was the question. The addendum actually, in some sense, admits that can always happen. even if you carefully plan your trial, you sit together, clinician, statistician, everybody involved, and you think hard about all the intercurrent events that can happen, sometimes you miss something, and that's perfectly okay, like COVID-19, okay? Nobody thought of COVID-19 when you planned the trial 3 years ago. That's still running. That's perfectly okay. I think then the addendum proposes a very pragmatic approach and has an explicit section saying emergent intercurrent events, what you should then do. And I think then you just say, okay, here is what we did, this is what we found, and here are the strategies we use, and then maybe you do it, you vary the strategies you apply or the assumptions, and just try to assess how robust is the result against these additional intercurrent events. I think very pragmatic. It's not, the expectation is not, I think you should be careful in identifying intercurrent events, but it's acknowledged that sometimes things just happen that you couldn't foresee, and that will not break the trial. You just need to make sure it doesn't dramatically impact the result.
Okay, thank you so much, Kaspar. I believe the other question we have already answered previously. It's quite similar. Maybe it's also related to COVID. So if we can move, we have no other additional question at this point. So if we can move to the next item.
Okay.
COVID-19.
Yeah, COVID. So COVID really, I think, illustrated the benefit of the estimand addendum, and maybe we can try to understand that a little bit better by thinking about what actually did COVID cause to our trials. Well, there are two main things. One is the healthcare system disruption. So patient missed visits, they missed doses, you had regional lockdowns, shutdowns, whatever, that patients could not travel to hospitals, and that made them miss visits and miss doses. And I think it's no surprise, that's very indication specific. If you are an oncology patient, and basically it's about that, it's about life or death, you take more risks to travel to the hospital and get your dose. If you are an Alzheimer's patient for whom going to the hospital always means your spouse has to go with you, you stay there for 2 days, you're in a trial of a drug, nobody knows it's working anyway, I think you'd be more inclined to just say, no, I mean, with this COVID, I don't do that. So I hope this illustrates that the frequency with which doses were missed and visits were missed might be very indication specific. So we have much more difficulties in neuroscience than in oncology, for example. Okay. And then there are COVID-19 associated events. For example, you use prohibited medications to treat COVID. You maybe have COVID. You might use medication to prevent COVID, or I don't know whether that's possible, but you treat COVID. You might die due to COVID. But if you are a cancer patient and you die, is it now because of COVID? Is it because of the cancer? Is it because of the treatment? Okay. But all these clinical events, these are precisely new occurring intercurrent events that nobody foresaw. But you have to deal with them. And the estimand framework gives you a framework to, in some sense, not easily, but in a structured way, deal with them. Okay. Go back to Yosemite. There were 4 intercurrent events identified related to COVID-19. Discontinuation of study treatment due to COVID-19, because patients had COVID-19, for example, or they couldn't travel because of COVID-19. Use of prohibited systemic treatment or prohibited therapy because of COVID-19. Missed doses that with a potentially major impact. So that's, I think, so it's not discontinuation, it's just missed doses and then they went back on treatment. And death due to COVID-19. So 4 intercurrent events that were added to the 2 that we discussed before. Okay. Now the question is, what is my, in light of the pandemic, what is my objective now? And I think for many of our trials, our position was that the pandemic will end one day, and we want to assess the effect of the drug in a world where there is no COVID-19 pandemic, because that will be the majority of the time the drug will be used. That is the world, hopefully, okay? So the trial objective and the primary estimand largely remain unchanged. That made us propose that for many, or here in this case, for all these intercurrent events, we propose a hypothetical strategy. Okay. Because if a patient dies due to COVID-19, and here, this is not an oncology treatment, this is treatment of diabetic macular edema. So if a patient dies to COVID-19, there's little discussion whether that could actually be diabetic macular edema. I think that's just not plausible. But if you are a fourth-line lung cancer patient, that's not so clear. And there you would not use a hypothetical strategy. There you would clearly attribute the death to the disease or the drug and not use a hypothetical strategy. But here, I think this is different. And I think here it can be well justified to just say we use hypothetical strategy for all these intercurrent events. Very short, but that led, then you basically just use MMRM, you take the data, what you have, and that implicitly applies the hypothetical strategy for all these intercurrent events. But that's for this indication and for this trial. And I think it's important that this needs a trial by trial, indication by indication specific consideration. If you're interested in the oncology context, the Oncology Estimand Working Group has written a paper, and that was actually quite intense experience, because we decided that we should provide the community with this information, and within 5 or 6 weeks we actually wrote that paper. But that was quite intense. But if you're interested, you can have a look, and there we make all this specific for oncology. Okay. So the next question is, very often the question is, and we already discussed this a little bit today, what type of activity should we apply the addendum to? Is real-world data in the scope? My position is that in terms of endpoints and in terms of whenever you think this framework is useful for you to develop your analysis, then use it. I think that there is no restriction to that. You might also have heard of the target trial framework in epidemiology. That's very, very related to the Estimand framework. And in some sense, it's a little bit a pity these have been developed independently. But they share the same goal, and they're very related. So I think the framework is useful in thinking about any endpoint, because very often you have intercurrent events, not just for time to event, also for a binary continuous endpoint, you have intercurrent events, and it's a useful framework to deal with them. The addendum says that you should for sure use it for endpoints that pertain to regulatory decision making. And typically, these are those endpoints for which we do type 1 error protection. But I think it's even useful for exploratory endpoints, PK, whatever you think of. So use it broadly. Nothing speaks against using it broadly. Which trials should we apply it to, or which type of studies? And I think we discussed this. So whenever you are interested in a treatment effect, then it makes sense to use it, be it for efficacy, safety, doesn't matter, PRO, it applies to all of that. The main focus is on RCTs, but it equally well applies to observational studies, single-arm trials, pragmatic trials, real-world data, whatever you think of. And it's just not, if it's useful, then use it. I think that would be my position. Same for data types, binary, continuous endpoint, longitudinal, time to first event, recurrent event. It equally well applies to all of them. I have not talked about so much today about missing data and the distinction between sensitivity and supplementary analysis, and because that's actually in the title of the addendum. It also talks about sensitivity and supplementary analysis, but we have dealt with this a little bit through the questions. But I'm more than happy to discuss further via email or in follow-up TCs if you're interested to learn more about this. I understand here we have a lot of clinical colleagues on the line. So I mean, that's all what we want to know always. What does it mean for me? And I try to summarize that a little bit. So I think we should always have the ambition to have a clear trial objective. We run multi-million-dollar enterprises, which these trials are. So we should be very clear what we are trying to achieve. And the estimand addendum gives us a tool to be more explicit and clear about the trial objectives. Okay. And for clinicians, I think be it in pharma, be it in regulatory agencies, I think it's important to recognize that developing an estimand requires discussion between all stakeholders and all specialities. So I mentioned that here so we need to discuss among sponsors and regulators that we agree on an estimand up front. It needs expertise from all the disciplines, statisticians, clinicians, regulatory, everybody involved. I think what we need to appreciate is that it might require more investment at the design stage, because we have these extended discussions, but it should avoid a lot of discussions at the analysis and filing stage, where we have 2 or 3 iterations of a lot of questions because we didn't, one stakeholder thought the objective implied this, and the other stakeholder thought the objective implied this. There is a famous example in diabetes where that even led to an ODAC or an FDA kind of public hearing, because sponsors thought they do this and the FDA thought the sponsors is doing that. And then at the filing, they discovered they're at odds, and that we want to avoid. I think so. That's what we're also experiencing, of course, in health authority interactions. We now discuss the estimand, of course, because that's the key. That's the treatment effect we're estimating. And something we are currently pushing through in our company, but that's a difficult enterprise, is we need to adopt this framework in all documentation. Synopsis, so we can go through the whole food chain if you want. Synopsis, protocol, statistical analysis plan, data collection, ECRF, and then CSR, summary of clinical efficacy, summary of clinical safety, clinical publication. Everything needs to adopt the framework and describe the estimand. We're not there yet, but that's what I think many companies are now working on. And also cross-company, like Transcelerate with all their templates, that they build this in their templates. Okay, so what's the impact that we see today? And just to mention that, I think Swissmedic was the first agency that approved the estimand addendum, maybe, I don't know, almost 2 years ago. And the FDA just did a week ago. So it's now also FDA formally approved, if you want. I'm not aware of all what makes these processes so different at different agencies, because ICH approved it long ago, but FDA now formally adopted the estimand addendum as well. So it's not like something you can do or you cannot. It's something that's in ICH, and now all health authorities have adopted, major health authorities. So what is the impact? And I think I can be very short here. If you're clear on the estimand, we are clear on which data we need to collect. We've discussed this in the context of treatment switching, treatment policy. You need the data after treatment discontinuation, because you just measure BCVA at week 52. You need to be more clear in terms of identifying intercurrent events. Why is data missing? Can we push back? Can we go back to the site and ask, why did the patient leave the trial? because that might lead to a different type of intercurrent event. Maybe we need more data than previously, for example, to model switching. One beauty of the concept is that different stakeholders might have different estimates, but that's perfectly okay. And the framework just adds transparency to that. Okay. It is a collaborative thing, multidisciplinary. I will not tire to emphasize that. That is very, very important in my opinion. And just to give you an indication, there is this very nice example from Novo Nordisk, where after the addendum, they started focusing more on retention, basically addressing this point. So they wanted to keep the patients in the trial, even if after they discontinued the treatment, to just get their endpoint value. And by some measures they took, they could increase completion rates from 90 to 98% in type 1 diabetes and from 17 to 90 in obesity studies. And you see here that just because we now have a framework, we can explain everything very well, even to patients, makes something like this possible. And that ultimately increases the quality of the evidence we are generating for our medicines. And here is just a summary of the impact on documentation. That's not even comprehensive because, yeah, they're missing some key documents even. But you see here, so many sections in our standard documents will be affected. The broader impact I see is that we get more agreement in clinical trials, we get more transparency, and hopefully we get more predictable assessment procedures so that we don't have these discussions at the time of filing, but we have them at the time of design of the trial. We also, and that was a promise from ICH, that we will reduce the total number of analyses because we will have more focused sensitivity analysis, but maybe not 15 of them, but just 3 of them for which we precisely understand what they mean. So that is our hope, that we need to provide less analysis. And I think that's an important point for us. We need a little bit of a shift of resources from the analysis time to the filing time, to the design stage. We need to invest more when we design the trial. More discussions. So finally, the addendum turned out to be highly useful for assessing the impact of COVID-19. I think that's very important and pushed the addendum. It requires a mindset change. We need to pre-specify more. We need to be more specific, more precise up front. We're all learning here. Everybody involved is learning and is making this high-level concept of the addendum concrete for drug development now. And I encourage everyone to work together within respective bodies, so within companies, within regulatory agencies, but also across. So we work a lot with regulatory statisticians, writing joint papers about methodology, about estimation methods, and making things concrete. For example, and I anticipate that will also happen for other functions like clinicians or safety or specialties. If you're interested to learn more, I have added here a few links. There is the official ICHE9 training material. So that's a very much extended version of what I was discussing today. There is a 3-minute intro video, which is actually very, very nice. I think it's also by Novo Nordisk folks who wrote a paper and then generated that video. Very useful. And just in 3 minutes, you get the essence. There is a webinar online targeted at non-statisticians. And there is actually an PSI webinar next week for estimates in oncology that you can still register for. So reach out if you're interested. There are cross-industry working groups. You find them on the internet if you're interested. And this is what I conclude with. This is something Frank Bretz came up with. So he was part of the ICHE9 working group. If you see here the estimate, if you reorder the letters, it gives you a mindset. And I think it's beautiful. And that's what it is. It is a mindset change. And I invite everyone to embark on that journey and that we develop better trials in the future. Thank you for your attention and happy to take more questions.
Thank you so much, Kaspar. So let's take the opportunity because we have one more question. So I believe the one that I said had been addressed, maybe it was not fully. So let me ask you here. What's the best strategy to address with intercurrent events in the COVID-19 clinical trials? Would it be population stratum or policy strategy? For example, in COVID-19 vaccines clinical trials, there is a discussion about the population stratum approach to address the problem of the intercurrent events.
Yeah, I think I would need more details to provide a qualified answer. If you're interested, I mean, principal stratification is by far, I think, the most difficult to grasp strategy that's put forward in the addendum. We in the Estimand Oncology Working Group, we have actually written a paper about the principal stratification strategy where we also a little bit discuss vaccines. So that could be a starting point. I think I would need more details to be able to say what, and I think it's not very often there is not a strategy that's correct and the others are wrong. It's all have pros and cons, and it depends on what you want to achieve, what your scientific objective is, clinical trial objective, and then from that you can derive which strategy to use. But without knowing more, I hesitate to provide here some kind of template answer.
Leonardo, não sei se responde a sua pergunta, ou se você quiser incluir alguma informação adicional para que o Kaspar possa responder, por favor, fique à vontade. Temos 4 minutos mais para a gente aproveitar esse tempo com o Kaspar e fazer mais algumas questões. Leonardo says that it was answered, Kaspar. Okay, so let's take advantage of these few more minutes. Any other questions? Leonardo has just made a comment. I think you have mentioned also that the E9 was adopted by FDA last week, right? Thank you, Kaspar, so much for your comprehensive and excellent presentation. I think you see that the participation of the audience was quite high. So for sure, it's in relation to your great presentation. I'm pretty sure that the guys here feel confident to reach you out in case of any other question they might have. I don't know if you want to say any final words. So let's end up our session today. Thank you all so much. Have you a good and rest of the day. Bye-bye. Obrigada a todos.
Thanks,
Tenham
everyone.
ótimo fim de dia.
Bye-bye.
Um abraço. Tchau, tchau.
Bye-bye.